As we enter iGEM's third decade, the focus has shifted from what we are doing with synthetic biology to who is driving it. Our vision of cell-free for all is grounded in the belief that synthetic biology should be universal, accessible to everyone, everywhere. Cell-free systems are an innovative tool that is accelerating the progress of synthetic biology. They are created by losing cells, which means the removal of their envelope and genome while keeping the expression mechanisms intact. In this way, any required biomolecule can be produced, only adding its genetic information and an energy solution, providing the vital resources. Constraints imposed by the cellular environment, like viability and metabolic burden through resource-intensive synthetic pathways, are no longer a problem in cell-free expression systems. Additionally, the lysate is dead and not able to reproduce. Biocontamination is therefore not possible, increasing the usability of these systems. This allows for faster design and testing of synthetic biology applications, like gene circuits to produce biosensors and vaccines. Furthermore, they can be utilized for educational purposes due to their easy handling, portability, and fast response compared to living cells.
However, there are challenges. One major issue is cost. Cell-free systems often require expensive reagents, particularly energy buffer components, and specialized equipment for preparation, making them less accessible to underfunded labs. Another problem is stability. As the components of a cell-free system need cryogenic storage, which is both cost-restrictive and impractical for the use in rural and less developed regions. This makes it difficult to broaden adoption, highlighting the need for innovations to reduce cost and improve stability in cell-free technologies. Bluebear Bio makes cell-free systems scalable, affordable, and easy to use, enabling more people to engage in synthetic biology.
We chose to prioritize social sustainability by developing a cell-free system that has the potential to remove accessibility barriers and enable their use worldwide, regardless of resource limitations. We wanted to address this challenge in two key ways. Number one, by the stabilization of cell-free systems at room temperature. This allows for storage and transport without specialized equipment or cold chains. Second, we wanted to lower the cost of the energy buffer by substituting the most expensive components without sacrificing its performance. We found our inspiration in tardigrades, also known as water bears, which are renowned for their remarkable ability to survive extreme environmental conditions, including desiccation. When faced with dehydration, tardigrades enter a state called anhydrobiosis, where they lose nearly all their water content and enter a dormant state. They produce specialized intrinsically disordered proteins, which form a glass-like matrix that shields cellular structures and components from damage associated with desiccation. This allows tardigrades to endure desiccation, extreme temperatures, radiation, and even the vacuum of space, returning to normal life functions once rehydrated. We set out to try to harness the power of tardigrade CAHS proteins to stabilize cell-free systems to eliminate the need for cost-intensive transport chains and storage systems. We constructed an inducible plasmid to co-produce tardigrade proteins alongside an autolysis gene from bacteriophage lambda, which allows the production of lysate with a simple freeze and thaw cycle instead of a more specialized process like sonication or physical lysis. The integration of the tardigrade proteins enables vacuum desiccation of the lysate and storage at normal conditions without the need for a freeze dryer or a -80° freezer. These modifications together offer an accessible and scalable expression system that can be created and used in even the most resource-restricted labs. Our results demonstrated that the lysate produced from strains that express tardigrade genes had an improved stability after lyophilization without additional protectants. Additionally, we stabilized the energy buffer using sugars and successfully desiccated the system using a conventional vacuum desiccator, eliminating the need for a freeze dryer. To further improve access, we proved that lysates produced with conventional tabletop centrifuges works just as well as with an ultracentrifuge. We also demonstrated that our desiccated lysate can be rehydrated with different water sources, including unfiltered water from the Danube. This not only means that it's accessible to more people, but also that our system can be used for point-of-care applications, such as testing water quality in remote locations with a biosensor. To prove our concept, we sent our desiccated cell-free systems to other iGEM teams via conventional postage systems. We eagerly await the feedback in the weeks leading up to the Jamboree.
For the second part of our solution, we conducted a thorough literature review and cost analysis to identify the most cost-restrictive components of the energy buffer, which were most promising to substitute. We then tested individual substitutions to assess system functionality. We decided on replacing purified amino acids with complex amino acid sources, such as yeast extract and tryptone. Next, we tried to replace phosphoglyceraldehyde with maltodextrin and hexametaphosphate. Nucleotide triphosphates were substituted with nucleotide monophosphates, and coenzyme A with its precursor, pentathenate. Replacement of multiple components simultaneously brought up a significant challenge: multivariable optimization with no available references in the literature for replacing all three components at the same time. To circumvent this issue, we decided on a design of experiments approach, optimizing the energy buffer by incorporating the most viable combination of these components, exploring the fitness landscape further.
Using design of experiment methodologies, a number of experiments were significantly reduced compared to traditional methods, such as testing one factor at a time or even certain machine learning approaches. This allowed us to address the complexity of the system and move closer to formulating the ideal energy buffer mixture. The optimization criteria were based on the standardized fluorescence of each reaction volume after introducing a known concentration of green fluorescence DNA template. We used a central composite design in our first DOE phase. One of our initial discoveries was that tryptone was less significant contributor as an amino acid source. This insight allowed us to exclude tryptone from further experiments, making our mixture simpler and more efficient. Additionally, from this DOE study, we could fit a precise quadratic regression model. This allowed us to predict how different variables interacted and gave us a better understanding of the optimum levels of each component. For our second central composite design, we use optimized conditions identified in the first experiment as the center points of the design. This iterative approach enabled us to refine our exploration even further by fitting another quadratic model for maximizing the standardized fluorescence and including standard deviation as a response to minimize. We could pinpoint the optimum recipe for our alternative energy buffer with a robust performance. We have promising variants, as you can see, where the price was reduced by 90% and the activity was only reduced by 25% and 45% for variant 1 and variant 2 subsequently. However, it is essential to consider the batch-to-batch variations of the lysate DNA yeast extract can impact drastically, as mentioned by Dr. Annabel Ers in our online meeting with him. The final mixture could support 75% of lysate activity compared to the canonical energy buffer described in the literature. This was a significant achievement, demonstrating that our novel energy buffer recipe could effectively replace more cost-intensive traditional ingredients.
Our experiments in Flask were successful, but we thought there was a room for improvements in yield and performance. To address this, we turned into bioreactors to see if our lysate production is scalable or not. After initial optimization of inoculum volume, we used 2x YTBG medium and high cell density fermentation. The results were promising, as the lysate yield nearly tripled, and there were room for further optimization. Next, we conducted simulation using SuperPro Designer to gain insights into an industrial scale process. Here, we identified the primary equipment needed, material flows, operational parameters, and the process order. Our economic analysis revealed that the process is feasible and potentially profitable. Finally, the enhanced scalability could also enable less variability and ensure reproducible results in our batches.
Our motto is cell-free for all, and our vision with this project is to democratize cell-free systems. This means that human practices are at the center of our project. We reached out to several potential stakeholders to understand their pain points and incorporate their feedback in aligning our project with our vision. We reached out to the iGEM community, researchers of cell-free synthetic biology, teachers, and biotech startups, grounding our project in actual, tangible boundaries. To assess accessibility constraints, we contacted various teams and communities, including Egypt AFCM, Synth Africa, Tech Chihuahua, and Tech de Monterrey, to understand the equipment and financial limitations faced by laboratories globally. Our discussions revealed insights into the specific challenges these teams could encounter when utilizing conventional cell-free systems. By gathering this information, we aim to tailor our project to better meet the needs of these institutions and enhance their ability to work with cell-free technologies. We sent our systems to the team of the University of Wageningen in order to properly assess the ability of shipping the stabilized lysate and energy buffer via standard means, without the employment of cold chains. To refine our research focus and understand the challenges and applications of cell-free systems, we engaged in in-depth discussions with field experts including Dr. Annabel Arce, Dr. Guzman Chavez, and Dr. Aurora Dupin. We also visited Invitris, a biotech startup specializing in cell-free systems, which provided valuable insights into industry practices and the cell-free startup ecosystem. This experience informed both our scientific approach and our entrepreneurship work, helping us align our project with real-world needs and opportunities in the field of cell-free systems. We found that many teams lack knowledge about cell-free systems. To address this, our project includes an educational component aimed at bridging this knowledge gap and raising awareness in society.
Cell-free systems, like the one we are developing, are particularly well suited for educational settings. We showcased our cell-free systems at schools, providing a safe and GMO-free hands-on exploration of biology's central dogma in real time without living cells. In total, we interviewed 4 teachers across Straubing and Regensburg and engaged with approximately 200 senior-level students in teachings and discussions. With the teachers' continuous input, we created a lesson plan. These loosely structured interviews revealed a significant knowledge gap surrounding synthetic biology. One of the major roadblocks were the time constraints and scheduling difficulties that teachers face when integrating SynBio into their curricula. Our cell-free systems shine here, as they can yield rapid results, perfect for a classroom setting. To engage students, we used visual aids and interactive quizzes with Mentimeter, allowing participation through their phones. We alternated between presentations and hands-on activities like a tardigrade hunt and using our 3D printed educational kit for GFP expression. The interviews with teachers were crucial as they provided insights that impacted the direction of our project and improved our lesson plans. These learnings refined our project through real-time feedback and improved our ability to communicate complex science effectively. Our campus events, on the other hand, gave us valuable feedback from researchers in a broad range of subdisciplines, helping us to further refine our project and strengthen our scientific idea. Together with our sponsor Sartorius, our team organized a vibrant community outreach event aiming to connect the campus community with synthetic biology and iGEM. For the first time, we presented our project to the campus community. By hosting a bake sale and a raffle with prizes donated by local businesses, we were able to create a closer connection to our community and fundraise additional funds for our trip to Paris. Attendees participated in a pipetting workshop and got hands-on with tardigrades using a microscope. The event was a hit, with lots of participation and support from the campus and local community. So we've got this project that we're super excited about. We saw all the potential applications for our technology and were keen to explore the entrepreneurial aspects of it, expecting to find a solid existing market. Cell lysate is already an established product on which we're lowering production costs, so it seemed logical that our innovations could market our product to a new niche that might not have been able to afford cell-free systems. We set out to carry out market surveys to discover our customer archetype, from which we received a low number of responses, but they still allowed us to gather some conclusions, the main one being that cost was a significant factor impacting the adoption of cell-free systems. We even validated the archetype with Team Tech Guadalajara, who shared their experiences of wanting to use cell-free expression systems, but were unable to do so due to the high shipment costs. We received prototyping funds from iGEM TUM to complete our vision and established working relationships with other startups operating in our field. Our business model is built on producing low-cost reaction kits, addressing an untapped segment of the market by enabling them to access revolutionary technologies. We're working towards a future where safe, accessible, affordable, synthetic biology tools are available to a broad audience of innovators, and a robust cell-free platform is an integral part of our vision.